Oh, there we go. Alrighty. Well, thank you so much for having me. So before I start, I'll thank the people. Whoop. Okay, here we go. Thank you. Okay, I'll thank the people that have done all the work. So these are current and former group members. I've really had the pleasure to work with an excellent team over the last couple of years. And I also really want to thank the Dubois and Lausanne, where most of the reconstructions were done that I'm going to show you today. All right, so I'll start with a simple example here. So CCMV is an icosahedral plant virus. And so like most viruses, CCMV has the following problem. It needs to find a way to package genetic material safely for transport, but then has to find a way to release it at the right point in time in order to infect the host. Right? And so the way CCMV does this is that once inside the host cell, CCMV detects a change in its chemical environment, and then in response to this change, the capsid here inflates. In fact, it undergoes quite a dramatic transformation here with the diameter increasing by about 10%. And then this inflated form is unstable. The capsid falls apart, and the RNA, which is on the inside, gets liberated, and the host is infected. All right, so if you think about it, this is a very sophisticated nanoscale machine. Right? It senses a chemical signal and then somehow translates this into this large-scale mechanical action. And so the point I want to make here is that we don't really understand how that works. So we do know what the chemical signal is. And if you compare these two structures here, you can sort of tell that there must be a set of translations and rotations of these capsid proteins involved that bring about this transformation. But that's really where our knowledge stops. Right? So we don't know if all of these motions happen simultaneously or in an asynchronous fashion. We don't know if there are any intermediates involved. We don't know if the icosahedral symmetry is maintained. We don't know any of this. And the reason why we don't know is because we can't observe this process, mainly because it is too fast. All right, so how fast would that process be? And so to this community, these are all things that are very well known. So proteins have motions over a wide range of timescales, right? So if you look at photoactivated cysts, you get these very fast motions here on femtosecond, picosecond timescales, and then you go all the way to motions on microsecond, millisecond timescales. For most proteins, the motions that are associated with functions are those of entire domains. And those usually actually would be kind of slow on this microsecond timescale. So as an example, if you were to ask what is the typical timescale associated with the catalytic activity of an enzyme, the answer would typically be microseconds. And this is also the timescale that we would expect for this process here, right? So really what we would need then to understand this process is a technique that offers microsecond temporal resolution. So right, you could do an optical pump probe experiment. You could try to figure this out, or small angle X-ray scattering and all of this. However, that wouldn't tell you the full story, right? Really what we want to know is, right, in this reconstruction here, every dot is an atom. And actually, to really understand how this thing works, we would like to know where each of these dots is as a function of time. So we really also would need something that has near atomic spatial resolution, right? And so a good technique to observe this process just simply doesn't exist. So, right, if we go through the time-resolved methods available to structural biologists that have atomic resolution, there's NMR. Actually, we do know a lot of these timescales from NMR spectroscopy. However, most of these experiments here actually are done at equilibrium, and the process we want to look at is far from equilibrium. And I would argue it's one of the defining features of life that it happens far from equilibrium, right? And so for out of equilibrium processes, NMR is many of orders of magnitude too slow. And then to many of you, ultra-fast X-ray crystallography is very well familiar, right? And there's beautiful experiments that have been done here. The only catch is that, of course, your experiment always has to happen within a crystal, and protein dynamics don't naturally occur within a crystal. And it's well known that actually these large amplitude motions tend to be hindered by the crystal packing, right? The process we want to look at, it's very hard to see how that would fit into a crystal. And then finally, there's cryo-EM, which also does exist in a time-resolved version. And so that there you get a time resolution of about milliseconds, right? Which is, again, 3 orders of magnitude too slow what we want to observe here, right? So in other words, then this process here it cannot be observed, right? There isn't a good way to look at this. And so then you could argue, right, if we're always limited to looking at static structures of proteins, then really understanding how proteins works must be incomplete, right? Another way to look at it maybe is to say we've gotten very good at obtaining protein structures, right? And now that we have so many, we can even use machine learning to predict protein structure, and those, of course, are amazing achievements. However, to really understand protein function, we need observations. And so this is now something that my group has done over the last couple of years. So what we've done is we've introduced a a microsecond time-resolved version of cryo-EM, which we think is hopefully going to be able to fill part of this gap. So here's how this works. So we take an ordinary cryo sample, so you have your proteins here embedded in this vitreous ice, and then we come in with a laser beam to very rapidly melt the sample. And as soon as the sample is liquid, you can then apply a stimulus to drive protein dynamics. And then as these dynamics unfold, we switch off this laser here that keeps the sample liquid. The sample cools down very rapidly. It re-vitrifies, trapping the proteins in their transient configurations in which we can then subsequently image them. Right? And so initially, when we wrote the first paper and tried to publish this on just a proof of principle of how this would work, it was almost impossible to get this published anywhere. So reviewers, I think, didn't really want to believe that this is possible. And fair enough, this sounds like a wacky idea, right? So if you do a cryo-EM, then you know that, what I'm telling you here is you're supposed to warm up this sample with a laser beam. If you do a cryo-EM, you know that you're never ever supposed to do this, because if you warm your sample up, it will crystallize, and then you have to throw it away. And then even worse, at this point here, the sample actually would have to cool so rapidly that instead of crystallizing, it becomes vitreous, right? And so we've actually measured the cooling rate that you need to achieve vitrification very accurately recently, and you get a rate here of 10⁶ Kelvin per second. So this is very fast, right? And so who tells me that actually my sample here would cool that rapidly? All right, so let me show you that this is possible. So this is how we do the experiments in the lab. So we have a transmission electron microscope here that we've modified for time-resolved experiments. So the laser beam here comes in from the left, bounces our little mirror, and then we direct it at the sample. The samples that we use are very typical samples for cryo-EM. So this is the specimen support here is a gold mesh that supports a thin film of gold that you can see has this regular pattern of holes here, and your cryo sample sits on top of this. And now we come in with a laser beam. This is a green laser. We aim it at the center of one of these grid squares here, and then we locally melt our cryo sample. Right? And so now the very first thing we did was we wanted to show that it's possible to melt and revitrify these cryo samples. So what we did here is we on purpose made a really bad cryo sample. So what you see here is a hole in the gold film, and you see a lot of contrast here, and the diffraction pattern tells us that this is actually hexagonal ice. So this usually would be a cryo sample that you wouldn't be able to use. Right? However, if what I'm telling you is correct, then the following should happen if we now irradiate that sample here with a 20 microsecond laser pulse. During the laser pulse, this sample should melt. It should remain liquid for the duration of the laser pulse. And then as soon as we switch off the laser, the sample should cool down so rapidly that it doesn't have time to crystallize, but instead the sample should become vitreous. Right? And so this is exactly what we see. So after the laser pulse, you can see the contrast here has gone homogeneous, and the diffraction pattern tells us that this is vitreous ice. So evidently, we've melted the sample, and it remained liquid, and then once we switch to the laser, the sample cooled so rapidly that instead of crystallizing, the sample became vitreous. Okay, so evidently it cooled fast enough. So the next question then would be, well, how fast did it cool? All right, and so with a time-resolved instrument like this is a question you can answer very rapidly. This is a simulation that we'll maybe look at first. So when you look at the red curve here, so this is the temperature on the sample as a function of time. And so you can see that when you switch on the laser, the temperature shoots up. It does so with a time constant of about 1 microsecond. And then the temperature stabilizes. So this is really nice for a kinetics experiment. And then once you switch the laser off, the sample cools down again with a time constant of about 1 microsecond and goes back to cryogenic temperature. Right? And so experimentally, we get very similar timescales. Right? So I'll explain in 2 words how this experiment works. So essentially, this is a pump-probe experiment here where this is a micrograph of the specimen support, and now we collected a diffraction pattern here from the center of the grid square. This is where we aim the laser beam, and now this is just a diffraction pattern here of polycrystalline gold, but we can use the diffraction intensities as a high-speed thermometer, right? So if we now record this diffraction pattern as a function of time with nanosecond electron pulses, and what I'm plotting here is the diffraction intensity as a function of time. You can see that once the sample starts heating up under laser irradiation, the diffraction intensity drops, then it stabilizes and it recovers, and we get very similar time constants here. All right, so these thin lines here, those are simulations for increasing ice thickness. And you can see that as you make the sample thicker, you add more ice, you add more inertia to the system, everything gets a little bit slower. But even for a very thick cryo sample here, the cooling time here is still something like sub 5 microseconds. All right, so now what you have to realize is that the time scale on which the sample cools down determines how rapidly you can trap these transient configurations of your proteins, right? So this cooling time here essentially dictates the time resolution of your experiment. So then based on these simulations and these experiments, we would conclude that with these experiments here, we should get a time resolution of 5 microseconds or better, which is about 1,000 times faster than what's been possible before with cryo-EM, right? Most importantly, this gets us, in terms of the time resolution, to this microsecond time scale where many of these relevant motions of proteins happen that we want to observe, right? So we can melt and revitrify these samples very rapidly with very high time resolution. The next question then would be, well, is a protein happy to go through this process, right? And this is what we looked at here. So here we prepared a cryo sample of apoferritin being a popular test protein to do these kinds of experiments on. And now we melt the sample, keep it liquid for the duration of the laser pulse, then switch off the laser beam to revitrify. And the question is the structure afterwards going to be the same as the structure before? Right? And so here's the answer. So on the left, I'm showing you a reconstruction here just from an ordinary sample. And so there we get 1.6 angstroms, and you can see this resolution, individual side chains, water molecules. On the right side, this is a reconstruction from a sample that we've put through this process. Right? We get about the same resolution, and within the resolution that we have, these two structures are identical. And that tells you two things. The one thing it tells you is that the protein is perfectly happy to go through this process. And the second thing it tells you is that our method doesn't seem to impose any fundamental limitation on the kind of spatial resolution you can get, right? So in essence, the resolution that you can get on a given sample with cryo-EM, you'll also get with our sample, plus or minus. All right. So then we decided that it would be important for our method to understand exactly how this flash melting works. And so this is an experiment that we've done to really understand this flash melting process. This is again a pump probe experiment. So this is our cryo sample here. We come in with a laser beam to melt the sample, and as we do so, we probe with a 2 microsecond electron pulse to get a diffraction pattern of our cryo sample. This here again is a simulation of the temperature evolution of this sample. This is a really, really thick sample, so it only reaches the melting point here after 14 microseconds. And now what we do is we record diffraction patterns here as a function of time. Right, so this is what the diffraction pattern looks like before the laser pulse hit the sample, so before time 0. And so I'm just showing the diffraction pattern here in the vicinity of the first 3 diffraction maxima of water. Right, and so then we switch on the laser beam, the temperature starts to shoot up, and then at 136 Kelvin, we cross the glass transition temperature of water. So above this temperature, water turns into a supercool liquid, right? And so if we record a diffraction pattern here at 6 microseconds, we just get a diffraction pattern of the supercool liquid. And so if we take a diffraction pattern at 8 microseconds, something interesting happens. We see these very sharp features here growing in, which tell us that the sample actually is starting to crystallize. So what has happened is in this temperature range here, we've crossed the so-called no man's land of water, where water is known to crystallize very rapidly. And apparently, during the passage here, we've picked up some crystallites, which now grow larger. And this is why we see these diffraction features growing in. And then at 9 microseconds, these features get more intense as these crystals grow larger. And then finally, we cross the melting point here. And so then if we take a diffraction pattern here at long times, then again, we get the diffraction pattern of liquid water close to room temperature. Right, so what we learn here is that during laser flash melting, our sample partially crystallizes. Now, that doesn't seem to do anything to our protein structure. However, it's something that maybe we would like to avoid. Right? You could imagine you have a loosely bound protein complex that might be ill-affected by this transient crystallization. Right? So the question essentially would be, is there a way to go more rapidly through this no man's land of water here? Right? And so far, the experiments that I've shown you were done with these just rectangular laser pulses. Right? So you switch on the laser beam, keep the laser power constant, switch it off. And the idea here is if we could add a very sharp initial spike to this laser pulse to actually get a higher heating rate, right? So essentially you would design laser pulses that look like this, right? Where I'm showing you different shapes here, and now the simulations show us that the narrower and the more intense we make the spike, the more rapidly we can cross this no man's land. And the question then is, well, can we cross it so rapidly that we don't crystallize anymore? Right? And so the way we look for crystallization is we come in with a 10 microsecond electron pulse in this time window here to check for crystallization. And now we can determine what fraction of the sample has crystallized as a function of the heating rate that we achieve here. And so this is the result. So this is the crystalline fraction as a function of the heating rate. And we can see that as we increase the heating rate, the crystalline fraction goes down, and that at a heating rate here of 10 to the 8 Kelvin per second, we are actually able to outrun crystallization. So now we can do an experiment where during the entire experiment we have no more crystallization, and the sample is only ever liquid or vitreous. All right. This flash melting process has a couple of other interesting properties, and I'll just show you one more here. So this is a reconstruction of the 50S subunit of the ribosome here from just a conventional cryo sample. And what I'm showing you here is the angular distribution of the particles. And what you see here is that there are these pronounced maxima here, and then other orientations that show up in white aren't populated at all. That actually is an effect that's very well known in cryo-EM. So during sample preparation, your proteins tend to adsorb to the air-water interface with hydrophobic parts of the protein surface, and this leads to these preferred orientations. It's actually a reason why a lot of cryo-EM projects still fail, because if your preferred orientation is too strong, if the number of views that you get is too limited, you cannot get a three-dimensional reconstruction, right? Evidently we're able to get a reconstruction here, but if I'm showing you the distribution here of the resolution, you can see that along these viewing directions here, the resolution is actually quite poor. So now, very interestingly, if we melt and revitrify these samples with the laser beam, we get something like this, where you can see that we've broadened this angular distribution, so we're starting to populate a bunch of these missing views, and our resolution map here has gotten more homogeneous. And we see pretty much the same thing for every single protein that we work with. For some proteins, the effect is very small. For others, it's similar to what I'm showing you here, but we do see an effect for every single protein. The effect gets stronger if we use one of these shaped laser pulses, so you can see that here we also get a massive improvement of resolution, so we go from 4.1 angstrom down to 2.9 angstrom under otherwise identical conditions, right? To put this in perspective, to get this kind of resolution here with such a conventional sample, you would have to record 18 times as long, and of course this then very quickly gets prohibitively expensive, right? And so we wondered whether we would be able to do even better, so this is what we did here. So the idea is the following. So you have your proteins here in preferred orientations attached to this air-water interface. What if we could do the following experiment? So we, in the vacuum of our microscope, we leak in water vapor to grow the sample thicker, right? So we deposit a layer of amorphous ice here. And the idea is that if you now melt the sample, once the sample is liquid, these proteins here now should have lost any memory of where this air-water interface was, right? So these proteins now should be able to rotate around freely, and if we then switch off a laser beam, we should be able to freeze out these proteins in scrambled orientations. And this is the result. So this, again, is a conventional sample. This is a sample that we've revitrified, and this is if we revitrified the sample after depositing an additional layer of amorphous ice. And what you can see is that we still have some preferred orientation here. So apparently some of these proteins were able to go back to the air-water interface and find preferred orientation again. However, this maximum now sits at a different location, very interestingly. And so evidently we're able to populate different views here that previously weren't populated. And so we've recently written this up, where also we try to understand how all of this works mechanistically. And we think this is a neat toolbox to overcome preferred orientation, which plagues so many cryo-EM projects. All right, so what I haven't talked about is yet how you would use these experiments and to look at protein dynamics, right? And this really all boils down to the question of what the stimulus here is, right? And you know, if you have a photoactive protein, you could certainly come in with a second laser pulse, and we've started doing experiments like this now. You could also, by just cranking up the power of this laser here, do a temperature jump experiment, and we've also started doing some of these experiments. I'll show you some data later. And then, of course, you could use photocage compounds, and we've heard a bunch about photocage compounds already today. So there's caged ATP, caged ions, caged peptides. In the most simple case, this would be a caged proton, right? Or in other words, a photoacid. And this brings us back to CCMV. So what we've done here is to trigger the dynamics of this plant virus is the following. We've prepared CCMV in its extended state. You can simply do this by raising the pH to 7.5. However, now we've prepared this sample here in the presence of a photoacid, right? And so now, before really doing anything else to our sample, before even loading it into the microscope, we irradiate this sample with UV light, and we release the photoacid to drop the pH to below 5, right? Why do we do this? Why do we irradiate the sample while the sample is still frozen? Well, this gets you around the limited absorption cross-sections and quantum yields of any caged compound, because essentially we can irradiate as long as we want and dissociate as much of this caged compound as we wish, right? So with this method, we can easily do pH jumps, you know, going down to pH 2 or something like this, right? So this virus now sits in this vitreous ice. It experiences a pH that has dropped below 5. At this low pH, it really wants to go to this contracted state, right? However, because it's sitting in this matrix of vitreous ice, it cannot react to this pH jump. The pH jump, even though we've irradiated for a very long time to get this large pH jump, will only become active once we melt the sample, right? So sure enough, if we do a reconstruction from this, from the sample here where we've already dropped the pH, we get a reconstruction of the extended state. For comparison, if we prepare a sample at such a low pH, we get a reconstruction of the contracted state. So all of these viruses here are ready to contract, and they will only start doing so once we melt the sample, right? So then if we melt the sample and then keep the sample liquid for the right amount of time, switch the laser beam off to revitrify, we should then be able to trap proteins or trap these viruses in partially contracted configurations. And sure enough, this is what we see in the experiment. So here we've melted and revitrified with a 30 microsecond laser pulse. And you can see that this reconstruction that we get here after 30 microseconds is right in between the extended and the contracted state. You can see this particularly well if I show you an overlay of these three reconstructions, where I'm now showing you a cross-section. You can see this blue 30 microsecond structure is in between the extended and the contracted state. So clearly, we were able to trigger these virus particles to start contracting, and then we frozen out the sample with the viruses in partially contracted configurations. Right? Now, if you look at this reconstruction, the resolution isn't so good. And this is not because there's anything wrong with our method. The reason really is that all of these particles contract at different speeds. Right? So really what you see here is sort of a motion blurred average. Right? Another way to look at this is to say, actually, there's a lot more information in there that we can try to get it with computational tools. And so what we've done here is we've mixed the data sets of this extended, this contracted, and this 30 microsecond ensemble and subjected these mixed data sets to a principal component analysis. Right? And I'm plotting the populations here as a function of these first two principal components. And what you see is that this 30 microsecond ensemble shows up neatly in between the extended and the contracted state. Now, this first component here, if you look at it, loosely corresponds to the change in diameter of these particles. So now what we can do is we can slice through this population here along this first component and then do a separate reconstruction for each slice in the hopes of getting more information of the trajectory that the system follows as these particles contract. Right? So for comparison, this is again the structure of this capsid here where I've highlighted the asymmetric unit, which consists out of these three proteins. And now we do reconstructions and try to extract molecular parameters from each of these slices, right? So what I'm plotting for you here is the particle diameter that we get as a function of the slice number. And you can see that for these first slices here, right? This is essentially the slices that correspond to the extended state. We get a constant diameter. These last slices here that correspond to the contracted state, we get a smaller but also constant diameter. And then in between, we get these slices that come from this 30 microsecond ensemble. And you can see that we have a wide distribution of diameters, which confirms our suspicion that all of these particles actually contract at different speeds. Right? And so on top of the contraction, these pentagons and hexagons that the capsid is made of rotate around their axis. And you can see that the pentamers rotated about twice the speed of the hexamers. Right? So this is now something that I wouldn't have expected. And then on top of all of this, these subunits here rotate around these axes drawn out as these black arrows. And you can see that the subunits A, B, and C all rotate at the same speeds. However, for these particles here that have contracted the most, subunit C has already completed its rotation, whereas A and B have only made it halfway. Right? So we get a very detailed picture here of how this contraction process actually happens. We can also build molecular models. So this is the extended state, this is the contracted state, and I can play a movie for you of how this contraction happens. So there's 2 points that I want to make here. You frequently see these movies where people have captured two states and they just do a morph, which essentially is a linear interpolation. And of course, strictly speaking, this is sci-fi, right? Because you don't know if you can interpolate linearly. This is clearly an example where you cannot interpolate linearly. But what I'm showing you here, this actually is reality. This is really how this process works. The other thing I want to mention is this is the only method that will give you these sorts of data. Right? That's the only way really to observe this process. And so now really what we've done is we've gone quite broad, looked at very different systems, at membrane proteins, very delicate proteins. And so it seems that this method really is quite general, should be applicable to a wide range of systems. There's one more thing I wanted to talk about very briefly before I run out of time. If I go back here, you can sort of see we've captured half of the contraction process. Why didn't we capture the rest? So if you have these samples here sitting in the vacuum of your electron microscope, of course, as soon as you hit these samples with a laser beam, they start evaporating, and ultimately they're gone. right? So how could we deal with this? And so we figured out that one way to get around this would be to cap these samples here with sealing membranes that prevent evaporation and keep these samples stable. And we figured out you can easily do this by depositing a vapor here of silicon dioxide onto the cryo sample from both sides. So we've built a vacuum deposition setup that allows us to do this. Now the trick with this is if you make these membranes too thin, they'll probably break when you hit them with a laser. And if you make them too thick, you lose too much spatial resolution. You can't get high resolution information anymore, right? And it turns out we can get away with really thin membranes, 1.4 nanometers here, right? These membranes are thin enough to get high resolution reconstruction. So on the left, this is again a reconstruction from a conventional sample. This on the right here is a sample now that we've melted and revitrified several times to keep the sample liquid for about 200 microseconds, and we only lose 0.1 angstroms in spatial resolution. So we can still get high resolution reconstructions even in these samples. This is only work for apoferritin. This is again a conventional reconstruction of the 50S ribosomal subunit, 30 microseconds. This is at 300 microseconds. Interestingly, this is again a plot of the preferred orientation of the particles where we have this very strong preferred orientation. The SEF star here is a measure of how much preferred orientation you have. So 1 would be a perfect isotropic sample. Everything below 0.8 is considered strong preferred orientation. Interestingly, in these samples here that we've coated with these membranes and revitrified, the preferred orientation goes away almost entirely. So our SEF star here goes to 0.99. Right? And we're currently trying to figure out whether this might be general and might be a way to eliminate preferred orientation entirely. Last thing I wanted to mention, this 50S subunit of the ribosome is, of course, a flexible system that undergoes motions at room temperature. One of them is this L1 stalk motion here, where I'm showing you the first principal component. Now, this is a temperature distribution on our sample, where the sample gets hottest in the center, and then this black line, this is where the temperature has dropped to 0 degrees Celsius. So we get a temperature distribution across the sample. Now, we would expect that in the hottest parts of the sample, we should get the maximum amplitude of motion here, and we should see less motion in the colder parts. So to figure out if this is the case, we sort our particles by temperature. And now what I'm plotting for you here is the amplitude of this motion here as a function of the temperature. And you see that at 30 microseconds here, we barely have any temperature dependence. Actually, it looks to be kind of negative, which probably is an artifact of how we extract the amplitude of this motion here. And so what seems to happen here is that at 30 microseconds, this is the amplitude of motion that corresponds to the temperature at which we've prepared the sample, right? The amplitude didn't have enough time to change, and then we see that at 150 microseconds, in the colder parts of the sample, the amplitude of motion narrows, and then at 300 microseconds, we get a pronounced temperature dependence here, right? And now, last thing I'll mention is you would think that this motion here should have a time constant of 100 nanoseconds. So why is this so slow? And this probably has to do with contacts at the base of this L1 stalk here rearranging as a function of temperature that limit the amplitude of motion. This is probably the time constant that we see here. Okay, I'm going to skip this slide here and just sum this up for you. So I walked you through the concept of doing microsecond times of cryome experiments. We get a time resolution of 5 microseconds or better. We think we should be able to go down to something like maybe 10 nanoseconds. We can get near atomic spatial resolution for suitable samples. Seems to be a neat trick to get rid of preferred orientation, a problem that makes a lot of cryome projects still fail. I've shown you this pH jump experiment here on this plant virus, and then also talked about how we can now access timescales of hundreds of microseconds. Really, we want to bridge the gap to the millisecond timescale where there's already traditional time-resolved cryo-EM. I haven't talked about this. There's essentially a trick to do these experiments in an optical microscope, which we think is an approach that should be more accessible to most people. I'd like to thank you for your attention. Be happy to take your questions.